Okay, can somebody confirm you can hear me?
Yeah, we can hear you.
Great. Before we begin, are there any questions? I don't think I'm showing my screen. Oh, good, the lecture file finished. Um, I'm a little confused on it. Am I sharing my screen?
No,
No.
you're not.
For some reason, I'm not able to get the normal buttons. There it is. All right. Um, so today is October 24th, and we're going to discuss Lab 6, Disc Diffusion and Antibiotics. Let me remind you that if you didn't view or attend last Tuesday's lab, I discussed the unknown project, and students often have trouble with the unknown project, and then I discover that they never viewed the unknown project's discussion. So to understand this project, you need to see the last lab. Um, all right, let's move on to lab 6, disk diffusion. Oh, let me also add that um, some students have been having trouble in the lab, and as we go later and later in the lab, meaning the later labs, have more difficult questions. And I am here to help you guys with the lab. So if you want verbal feedback, ask me questions during the lab. And if you have questions, make sure you're on um, early, because when the last student logs out, I'll log out as well.
I have a question. The last lab you said you were going to be emailing us our unknown number.
Uh-huh.
Um, but I haven't gotten one yet, and I was just wondering if
You
when
won't
you're
get that until next Tuesday.
okay, thank you.
But good question. All right, Lab 6, Disc diffusion, evaluation of disinfectants and antiseptics, and antibiotic effectiveness. There is some rating, as usual, from the textbook before you do the lab. In today's lab, we'll have some supplemental materials, including a video clip and a virtual lab. The learning objectives are: one, identify the type of chemical agent and the active ingredient used in various disinfectants and or antiseptics. Two, evaluate the effectiveness of disinfectants and or antiseptics using the disc diffusion method. Now, the disc diffusion method is also called the Kirby-Bauer method. Three, determine the mode of action of various antibiotics. Four, evaluate the effectiveness of various antibiotics using the disc diffusion method. Five, list variables that influence antibiotic disc diffusion susceptibility testing. And six, be able to define the following terms: sterilization, disinfection, disinfectant, an antiseptic, a bactericidal agent, a bacteriostatic agent, an antibiotic, a spread plate, and the zone of inhibition. Any questions about any of that? All right. So in evaluation of disinfectants and antiseptics, the first term is sterilization, which eliminates all viable organisms, including endospores. Disinfection refers to the destruction of vegetative cells, but not necessarily endospores. When we use something on an object to disinfect the object, we call that a disinfectant, a chemical agent applied to an inanimate object. An antiseptic is a chemical agent applied to living tissues. For example, if I were to use bleach on my toilet, it would be called a disinfectant. But then again, if a cat were to get injured and I were to treat it with bleach, the bleach would be called an antiseptic. Usually antiseptics are not as harsh to living skin as disinfectants, because obviously bleach would be harsh to the cat's skin. And oftentimes antiseptics aren't as strong as disinfectants, but that's not always the case. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is inoculated with a particular bacterium. Paper discs are then saturated with different chemical agents and then applied to the surface of the agar. The chemical agent diffuses into the agar, establishing a concentration gradient of the chemical around the disc. You then incubate the plate overnight at 35 degrees, and then you look at the bacterial growth pattern around the disc. If a zone of inhibition appears around the disc, an area with no bacterial growth, the chemical is assumed to be effective, at least for that particular bacterium. Conversely, if there's no zone of inhibition around the disc, the chemical is assumed to be ineffective. Now, this is effectiveness of the chemical. It is not sensitivity of the chemical. We'll talk about sensitivity a little bit. This table is telling you the different chemical agents, their general action, and their preferred use. I think the only ones we'll really talk about are alcohols, like ethanol is an alcohol, iodine and chlorine, like chlorine bleach or halogens, heavy metals. I guess we'll talk about that. Hydrogen peroxide is a peroxygen. The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. Various other factors affect the zone size, including the concentration of the chemical. Generally, the more concentrated the chemical, the larger the zone of inhibition. The molecular weight of the agent. Generally, the larger the molecular weight of the agent, the smaller the zone of inhibition. And the density of the bacterial inoculum. It depends on the density we're applying, but if you're using a specific dose and you increase the density, generally you would decrease the size of the zone of inhibition. Therefore, the only assumptions that may be concluded to the lack of standardization of these variables is you need to standardize the variables. Evaluation of antibiotic effectiveness. Antibiotics can be categorized as bactericidal if they kill the susceptible bacteria or the bacteria cells. They are categorized as bacteriostatic if they inhibit the growth of the bacteria, but do not kill the cells. When a bacteriostatic antibiotic is used, the immune system is used to assist in the elimination of an infection. Because once you stop applying a bacteriostatic antibiotic, if the bacteria are still around, the bacteria may continue to grow. Any question about any of that? And yes, we do have some antibiotics which are bactericidal and others which are bacteriostatic. I only know of a few bactericidal. Penicillin G, at least generally, is bactericidal. The Kirby-Bauer, or KB, disc diffusion method, also called the disc diffusion method, is the most common method for antibiotic testing. The results of such testing can help physicians in choosing which antibiotic to use when treating a sick patient. Bacteria can be isolated from the patient, spread on an agar plate. You allow the bacteria to be absorbed by about 5 minutes, meaning the liquid to be absorbed about 5 minutes, and then you put disc impregnated with an antibiotic on the surface of the agar. The disc will then diffuse the antibiotic around the disc, creating a concentration gradient. You then allow the plate to grow for 18 hours at 35 degrees in an incubator. If you have a zone of inhibition, then you can see, let me blow this up, like this, showing you a clear zone of inhibition right there. Then you can say that at least the bacteria are somewhat affected by this antibiotic, and there's a less of a zone of inhibition, and maybe these two have no zones of inhibition. So these would be an antibiotic which would not be effective on this bacteria. So you need to have the concentration of the antibiotic high enough in the area around the disc to get a zone of inhibition. And you do not know from this zone of inhibition if the bacteria have been killed or whether it's acting as biostatic, meaning the bacteria are just prevented from growing. So when you see a zone of inhibition, you just say the bacterial growth is inhibited. You don't know if it's killed or simply the bacteria's growth is stopped. The inhibition zone size is dependent on several factors: the diffusion rate of the antibiotic, the degree of sensitivity of the microorganism, and the growth rate of the bacterium. Table 6.2 lists the procedure required to standardize these variables when performing the Kirby-Bayer disc diffusion method. I don't know why that came up. Well, I tried to clear it. There it goes. Now I lost my mouse. So first you inoculate a nutrient broth using several bacterial colonies on a that you obtained from a non-selective media, and the bacteria should be no more than 24 hours old. You grow the broth for less than 24 hours and adjust the broth turbidity to 0.5 McFarland units. This would be approximately 10 to the 5th organisms per mil. You then inoculate the bacteria in the broth using a cotton swab at room temperature onto a Mueller-Henton agar plate. We always use Mueller-Henton agar plates because the media itself can affect the zone of inhibition size. And the Mueller-Henton agar plates are always between pH 7.2 to 7.4. And the depth of the agar is always 4 millimeters thick because the depth of the agar can affect the diffusion of the antibiotic in the disc. The greater the depth, meaning greater than 4 millimeters long, the smaller your zone of inhibition. And then the diameter of the plates are always 150 millimeters long. There's a specific technique of swabbing the plates. We'll talk about that. What you want is the bacteria to grow as a lawn of bacteria. You then allow the bacteria to be absorbed by 5 minutes because it's in a liquid. You want that to be absorbed. And then you apply the discs containing the antibiotic at least 24 millimeters apart. So you apply no more than 12 discs per 150 millimeter Mueller-Henton plate. And that's sort of shown up here. If you count those, there will be 12. We actually have a tool that dispenses them all at once in that pattern. So the antibiotic discs are plated 5 minutes after you plate the cells, but no longer than 15 minutes after you inoculate the cells. You then allow the plate to sit no more than 15 minutes and then stick them in a non-CO2 incubator for 16 to 18 hours at 35 degrees Celsius. If you grow them longer, you will have a smaller zone of inhibition than at 16 to 18 hours, and always at 35 degrees also. I think the temperature can affect the size of the zone of inhibition also. I'm not sure about that one though. I've never done it anything at 35. After incubation, the incubation zone size are then measured in their diameter using millimeters. A bacteria is considered to be sensitive or resistant to a particular antibiotic depending on the table set up by the National Committee for Clinical Laboratory Studies, which is often abbreviated NCCLS. Here we're seeing the table for Staphylococcus aureus, for the antibiotic penicillin. If the zone of inhibition has a diameter less than or equal to 20 millimeters in size, we say that Staphylococcus aureus is resistant, and that's penicillin G, by the way. If, on the other hand, the diameter of the zone of inhibition is greater than or equal to 29 millimeters, then we say Staphylococcus aureus is sensitive to penicillin G. And if the zone of inhibition is in between 20 and 29, meaning 21 to 28 millimeters, we say that the Staphylococcus aureus is intermediate to that antibiotic, in this case, penicillin G. And this one, if we measure it here, that measures 20 millimeters, maybe 19 and a half, which means that this strain of Staphylococcus aureus is resistant to penicillin G. Any question about any of that? So to determine the sensitivity or the resistance, you actually have to grow up the zones of inhibition, measure it in millimeters, and then compare it to a table set up by the NCCLS. And then look at the table to determine is it resistant, intermediate, or standard. Sensitive. All right, any question about any of that? So right there, can you say that that antibiotic is exhibiting to this bacteria? The bacteria is sensitive to that antibiotic? Can anyone state that? And how about that one? Is the bacteria sensitive to that antibiotic? Once again, you cannot know. You'd have to measure the size of the diameter and then look it up in the table. And I can't read what antibiotics those are, but it's most likely this one is sensitive because that's the largest, but that doesn't necessarily mean it, because this could be a very small antibiotic, in which case it'll have a larger zone of inhibition. And that could be a very large antibiotic, and it'll have a smaller zone of inhibition. So this one could be sensitive, and that one could be resistant. You don't know until you measure the diameter and then look it up in the NCCLS table. I can tell you that one is resistant, and that one is resistant. Any questions about any of that? All right. So know the terms of this lab. You can ignore the references. While you're not going to be physically performing any of these activities, you will be held responsible for how to interpret the effectiveness, of antibiotics using the Kirby-Bauer disc diffusion method. For procedure 1, we're preparing a spread plate. So view the video clip. I'm preparing a spread plate, and then review the steps for using aseptic technique when preparing a spread plate. Let me go ahead and talk about this. We're going to pretend that this is a plate of antibiotics, so that this will be the auger right here. And we have a tube, which I guess I can use. I'll pretend this. I'll just not touch it because I want to keep this. Can you guys see that? You can't. Could you see this? Yes, you could. Okay. So what you do is you get a cotton swab. We're going to pretend this is a cotton swab and dip it in the solution after it's been mixed, and then touch the sides of the tube. I'm not going to do that because I don't want to, this isn't sterile and I drink out of this cup. Usually what I do is I take this cup and cap it and take it in the car. But anyways, touch the inside of the tube so that the swab is not dripping. I'm doing the outside because I don't want to touch the inside. And then you take the cotton swab and then you streak it everywhere along the plate. Trying to go everywhere. I'm probably not getting everywhere. And then you turn the cotton swab a quarter of a turn. I don't know if you can see that or not. You can see my hand moving. And then you turn the plate a quarter of a turn. And then you cotton swab the plate once more, going everywhere. When you're all done, you then turn the swab another quarter of a turn, and then turn the plate a quarter of a turn. And then you swab the plate everywhere. The idea is to get the bacteria to grow everywhere on the plate. We call that a lawn of bacteria. And you do that 3 or 4 times. So this does do it 3 times, all right? And then at the very end, you take your cotton swab and you move along the edge of the plate. Because when you're swabbing, you frequently miss the edge. Like when I'm going like this, I might not go, you probably can't see it. You definitely can't see it. You might miss the edge. So you take the cotton swab and you go all along the edge. And that's to get the bacteria to grow on the edge as well. And that's to get a lawn of bacteria where bacteria will grow everywhere. I once had a student when I was in the lab, and she went doop, 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 doop. And that was it. And then she got her plate and she had these big holes where there were no bacteria showing. And she had a couple of paper discs in there. And she said, well, how can I measure the zone of inhibition? I've got these big holes in there. And I said, well, you didn't get the bacteria to grow on a lawn. You either have to redo it again, or you can use your lab partner's results. And the next time you do this, make sure you swab the plate everywhere 3 times so you get a lawn of bacteria. You then put the cotton swab in a container or biocide. Allow the plate to sit plate side up for 5 minutes. That's to allow the liquid from the cotton swab to be absorbed into the plate. And then put on the disc, which you could use forceps, make sure that they're at least 24 millimeters away from each other, or we have a device which actually pumps down 12 of them at the same time, being spread 24 millimeters apart. You put that down between 5 minutes and 15 minutes after you swab the disc, the plate, and then you let that sit for 5 to 15 minutes, and then you put it in the incubator upside down So I guess we'll just flip it because it needs to be in the incubator upside down. We always grow plates in the incubator upside down. And then you let that grow for I believe it's 16 to 18 hours. Let me look on here. 16 to 18 hours at 35 degrees Celsius. And then you measure the zones of inhibition using a millimeter ruler. Evaluation of the antibiotic effectiveness. The following video clips complete the virtual lab. Oh, sorry. View the following video clips and complete the virtual lab. So there's a little clip on preparing a Kirby-Bauer diffusion test, and then a little clip on interpretation of the results of the Kirby-Bauer diffusion test, and then the virtual lab, which maybe I should go to the worksheet. So I've got a note saying use the new link. I've got the link here. Looks like I don't have the link here, so let's go here. It'll take a little while to download. Hopefully it won't take too long. I think it's already open. There it is there. All right. Description, steps, credit, and start. Let's click start. This is showing you the first plate. Take your little ruler and then measure the diameter. And that looks like TE is maybe 17, maybe 18, something like that. And TE 17, 18 means it falls there. If it clicks green, that means you got it right. If you click there, that's incorrect. Click there, it's incorrect. Streptomycin, incorrect. That's correct, and that's incorrect. Okay. Yeah, streptomycin there. So that's about 18. Yeah, it's greater than 15. So that's how you do this. Now to get the second plate, you need to hit the menu and restart. And there's the second plate, which is Pseudomonas aeruginosa. There's the zone of inhibition. There's a zone of inhibition. There's a zone of inhibition. To get the third plate, you hit the menu again and hit restart. And there you can measure the zone of inhibition for VA right there. This looks like Staphylococcus aureus. This looks like it's MRSA, multi-drug resistant Staphylococcus aureus, because it's resistant to all of the antibiotics except for VA, which is probably vancomycin. And then to get the first plate, you can hit the menu and then the restart button once again. So that's how you do this. Any questions? All right. Let me close that down. Using the standards required for the Kirby-Bauer susceptibility test, an E. coli culture is prepared using a spread plate technique. Antibiotic discs are placed on the surface of the agar and incubated overnight at 35 degrees Celsius. Below are the results for the results along with the NCCLS zone standards used for interpretation. So here we have ampicillin. Let me shrink this down a little. And we have the measurement here for the ampicillin zone of inhibition. I'll tell you this one, and it's reading almost 15, a hair less than 15 millimeters. So more than 14, like I said, almost 15. And how you read the caliper is you do not read it from here to there. That would be like 9.5. You actually read it from 0 to 0. And that's why I'm reading almost 15. And then we take a look at the, oh, this is E. coli. I forgot to mention that. E. coli for ampicillin. And then the NCCLS table for E. coli and ampicillin. And I believe I said 15. And that would mean that it falls within the intermediate range, 14 to 16. So I'll actually fill that in for you. 15, interpretation is intermediate. Any questions about that? All right, do that for cephalothin, gentamicin, and tetracycline. Measure the size of the zone of inhibitions, and you got that right here. Measure it there, from here to there. And then take a look at the table and decide, is it resistant, intermediate, or sensitive. And then for gentamicin, the same, and then tetracycline, the same. We're always using this table. We're not using that one. And then fill in this table. Any questions about, I think that's question 2. Yep. And then for question 3, list the variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bauer diffusion method. You can get those from a table in the lab module. Then go to question 1. My apologies, the numbering is repeating. Let me look and see that that's actually the case for you guys. Okay, I redid the numbering for you guys. I don't know why I don't have it changed here, but well, let's go ahead and use Canvas. So that would be question 3. For question 4, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? 5. What would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? Mention what will happen to our test results. 6. What would happen if we increase or decrease the concentration of the antibiotic in the disc? Be specific in your answer. 7. Antibiotics can be bactericidal, that is, they kill the microbes, or they can be bacteriostatic, they inhibit the microbial's growth but do not kill the cells. When observing a zone of inhibition around the disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? This is a thinking question, and students frequently get it wrong the first time. Okay, so you'll need to think about this, and hopefully you'll get it right the second time, because most of you will get it wrong the first time, but think about it. This is a challenging question. Unfortunately, most students do get it right the second time. 8. When performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning the same kind of media, the same agar death, the same age of the culture. Why? So give your explanation there. 9. Are there other substances besides antibiotics that we could evaluate using this method? We're talking about the Kirby-Bauer disk diffusion method. For some reason, my mouse isn't working. Ten, does the presence of a zone of inhibition necessarily mean that the bacterium being tested is sensitive to that antibiotic? Explain your answer. And I need to update this page. I got to put in one more question. 11. From the size of the zone of inhibition of chloramphenicol on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to the antibiotic? And then for chloramphenicol on P. aeruginosa, is the bacteria sensitive, intermediate, or resistant to the antibiotic? Any question about any of that? Oh, I'm seeing it there now. Nope, it's not here. Oh, I see what the trouble is. So let me update that and put on question 11 and I'll make it available to you. All right, begin with the lab. Like I said, I'm adding a question, so you might hold off on downloading the worksheet, but you can start working on the lab other than the last question. Let me stop this. Are there any questions before we go